So, good afternoon, everybody. My name is Chrislyn Particka, and I'm the host for today's webinar. Our topic today is viral diseases transmitted through nursery stock in the East, grapevine leafroll disease, tomato ring spot, and grapevine red blotch, with Marc Fuchs, Mizuho Nita, and Annemiek Schulder. Marc is originally from France, where he received his master's and PhD degrees. He has worked on viruses of fruit crops and vegetables over the past 30 years, developing detection techniques, understanding spread, and investigating management strategies. For the past 7 years, he's been an associate professor with the Department of Plant Pathology and Plant Microbiology with Cornell University, based at the New York State Agricultural Experiment Station in Geneva, New York. Mizuho is an assistant professor and extension specialist of grape pathology at Virginia Tech's Alson H. Smith Jr. Agricultural Research and Extension Center in Winchester, Virginia. His academic interest is in applied plant pathology and plant disease epidemiology. At Virginia Tech, he engages in a range of research and extension activities, including exploration of cultural and chemical means of disease management, fungicide and fungicide alternative testing, variety susceptibility evaluation, and development of novel diagnostic tools. Annemiek is an associate professor in the Department of Plant, Soil, and Microbial Sciences at Michigan State University. The main goal of her program is to develop environmentally sound and economically sustainable methods of disease control in small fruits and grapes. Her current research involves the morphological, pathogenic, and genetic characterization of pathogenic fungi in blueberries, cranberries, and grapes. Remember that as a participant in today's webinar, we ask that you keep microphones muted if you have them. If you are having any technical difficulties, please type a note in the chat pod over on the right side of your screen, and I'll do what I can to help. If you have questions, please type them in the chat pod as well. We'll take a break during the course of the webinar between each speaker to address questions, and then hopefully we'll have a little bit of time at the end as well. So Marc, with that, I'm going to let you get started with your presentation.
Thank you very much, Chrislyn. Let me extend my gratitude not only to you for hosting this webinar, but for Team Martinson to put it together. Good afternoon, good morning, or good evening, good night, wherever you might be. Thanks for participating. Today, there are more than 70 different viruses that have been described in grapes worldwide. These viruses, or at least most of them, have various impacts on vigor, production, and fruit quality. They also have quite a diverse range of distribution on this planet. And in the Northeast, there are 3 major viral diseases that are of concern to the industry: Leafroll, tomato ring spot, and red blotch. And Leafroll is likely the most widespread disease of economic importance to the Northeast, being said a viral disease of economic importance. So the goal of my presentation today is to provide an overview of what we know today on the biology and ecology of these 3 major viruses. The first one is Leafroll that I'm going to touch upon today. This is a shot of a Lemberger vineyard in the Finger Lakes region of New York State, where you can see that most of the vines have a very dark red canopy, indicative of Leafroll disease. And looking at the distribution of the diseased vines, where you see that almost every vine in that vineyard is symptomatic, there is little doubt that the virus came through the planting material. Unfortunately, either the rootstock or the sign, or both the rootstock and the sign, when this vineyard was established, were infected with Leafroll. A typical symptom on a red-berried variety is, again, this reddening very early on in the growing season, nothing to do with reddening as associated with senescence. And very often the red leaves would show a downward rolling or cupping of the leaf blade, while main veins very often remain green. In white-berried varieties, as shown here in the Chardonnay, you see a diseased vine on the left in comparison to a healthy vine to the right. And here, disease symptoms are less conspicuous. And you can see an overall chlorosis of the leaf blades accompanied by a cupping. And these type of symptoms are very difficult to identify in a diseased vineyard. But more importantly than foliar symptoms, leafroll can have a dramatic impact on yield and fruit quality, as illustrated here in this comparative slide where you see the production of a healthy Cabernet Franc to the left and a leafroll disease Cabernet Franc to the right. Look at the less clusters on the diseased vine, and more importantly, the color of the berry skins on the diseased vines, most of which being a very pale blue or even pinkish color compared to the dark blue that you would anticipate from a healthy Cabernet Franc. Interestingly, when Tim Martinson measured Brix at harvest in 14 different Cabernet Franc vineyards in the Finger Lakes of New York, and the 14 different Cabernets vineyards are labeled A through N on this graph, and he plotted the differential in Brix between leafroll versus healthy. You can see differential varying from 0.5 to close to 4 units, which is a great indication as to how leafroll will affect fruit chemistry. What Tim also did is prior to harvest, a few weeks prior to harvest, he similarly monitored sugar content in Cabernet Franc and basically documented that leafroll delays fruit ripening as measured by Brix accumulation by 2 to 3 weeks upstate New York. And more importantly, if you look at the end product, this is again a leafroll in a healthy cluster compared side by side. You might notice a differential in anthocyanin content and coloring of the berry skins, with the leafroll affected being much lighter in color, and the end product is quite telling, with a poor quality beverage that you would obtain from leafroll infected vines, here shown Pinot Noir. Leafroll viruses, or at least most of the leafroll viruses, are transmitted from vine to vine or vineyard to vineyard by insect vectors from the family scale insects or mealybugs. In New York, we are fortunate to deal with only 3 identified vectors: the brown scale, the cottony maple scale, and the most important being the grape mealybug. So this is basically a brief summary as to what is known in terms of the biology and ecology of leafroll. Now I'm going to move on to the next disease of importance to the Northeast industry, tomato ring spot virus. Tomato ring spot virus is a very destructive virus. It's one of the most, if not the most destructive virus of vines in the Northeast, particularly on own-rooted hybrids, as shown here in the Vidal vineyard. And you see all the missing vines, or almost dying, completely dying vines in the forefront of this vineyard, and these vines are dying or have died because of the presence of tomato ring spot virus. And you can see also the distribution of these dying or dead vines that is, very patchy. And the reason it is patchy is because the virus is transmitted by vine to vine by a soil-borne ectoparasitic dagger nematode called Xiphinema americanum. And you have one specimen shown here on this picture. And the nematode, if you look at the mouth end of the specimen, has a stylet that will act like a needle of a syringe, by which the nematode will pierce the root cells and suck empty the cell content. And this is how the nematode will feed. and by feeding, it might acquire the virus, and moving from a diseased vine to a healthy vine, it will probe again on the roots of a healthy vine, therefore transmitting the virus. In terms of foliar symptoms, tomato ring spot virus is inducing some mosaic and chlorotic areas of leaves that are much smaller in size compared to healthy leaves. But again, more importantly than foliar symptoms is the effect of tomato ring spot on production. And here you can see the effect on the hybrid Cholois, where it is quite obvious that the virus induces shot berries, even-sized berries on clusters if there is any production. The situation is as dramatic on Vidal, where you can see the dramatic yield reduction illustrated by this comparative slide showing a cluster on the left from a tomato ring spot virus-infected Vidal and a healthy one to the right. So this basically wraps up what I had in mind to speak about tomato ring spot in terms of the biology and the ecology of the disease. I'll move on to the third one, being red blotch. And this is an overall view of a red blotch disease Cabernet Franc vineyard, where you might identify leaves that are red in appearance. And the reddening is, unlike Leafroll, you have these blotches or specks of reds, and the intensity of the reddening varies from very pale red to very dark crimson red. And either the full blade is symptomatic or only certain segments of the leaf can be symptomatic. On Pinot Noir, we have similar blotches and red specks. And in most red varieties, symptoms are very similar to those that I have just shown on Cabernet Franc and Pinot Noir. On white-berried varieties like Chardonnay, symptoms are again very different and are illustrated by some chlorotic areas of the leaf blade that can eventually coalesce later in the season, and these areas might become necrotic. Again, more importantly is the impact on fruit quality and yield, and you can see again here, as illustrated with Leafroll previously, the dramatic effect of red blotch on the production of Pinot Noir, where the clusters show very light-colored berries, indicating much reduced level of anthocyanins in berry skins of the infected clusters. The jury is still out there in terms of yield, but yield depressions have been clearly documented in certain vineyards affected with red blotch. Recently, the 3-cornered alfalfa tree hopper has been shown as a vector of potential epidemiological significance for red blotch. Now, if I try to wrap up our knowledge of the 3 major viruses of importance to the Northeast, Leafroll, Ringspot, and red blotch, Leafroll is caused by 5 different viruses. And these viruses are named grapevine leafroll-associated virus 1, 2, 3, 4, and 7. What is interesting, only 3 out of the 5 leafroll-associated viruses, 1, 3, and 4, are transmitted by mealybugs and or soft-scale insects. And as you can see, there are many different species of mealybugs and soft scales that have been documented as capable of transmitting Leafroll from an infected vine to a healthy vine, or from an infected vineyard to a healthy vineyard. In terms of ring spot, tomato ring spot is transmitted from vine to vine by the dagger nematodes, and there are 3 different species that are known to be vectors of tomato ring spot virus. Now, one thing to keep in mind is soil erosion or transfer of soil from one site that is infected or infested with the nematode that carries the virus to another site might be another means of transfer of tomato ring spot. Red blotch is an emerging disease for which the virus was identified in 2011, so very recently, and the alfalfa, the three-cornered alfalfa tree hopper, has been documented as a vector of this virus. So in conclusion, I hope that I was able to demonstrate that Leafroll, tomato ring spot, and red blotch can have severe effects on not only on vine vigor, but also yield, fruit quality, and the productive lifespan of vineyards. And I will not expand on the economic impact of red blotch and Leafroll, as this will be the topic of another webinar in the coming days by Shadi Atala. However, by teaming up with Shadi and Miguel Gomez, we have documented that Leafroll will impact Cabernet Franc by 9,000 to 16,000 acres over a 25-year lifespan of a vineyard, whereas the red blotch incidence or economic impact will vary from $5,000 to close to $40,000 per acre. You know, again, illustrating that these viruses have tremendous economic significance. What we learned also is that none of these viruses, the three we were talking about during this presentation, Leafroll, tomato ring spot, and red blotch, are known to be mechanically transmitted in vineyards, like the 70 others that have been described so far in grapes. This means that pruning, suckering, trimming, hedging, spraying, or harvesting will not move any of these viruses from an infected vine to a healthy vine, or from a diseased vineyard to a healthy vineyard. However, we have to keep in mind that some of these vectors are very tiny and very light and can be moved from mechanical activities in vineyards from one location to another. I hope I also convince you that symptoms can be sometimes typical of any given virus disease. However, they are not always reliable for diagnosis. And this is because there are so many other compounding factors that can interfere with a reliable diagnosis. And one good example is, I always tell growers or my colleagues that prior to the discovery of red blotch, I had believed that I was capable of identifying reliably Leafroll in any diseased vineyard. Well, guess what? After the discovery of red blotch, I will not put a reliable diagnosis again, ever, whether it deals with Leafroll or red blotch, because the symptomatology of the two diseases can be very similar, if not identical in some cases. One thing to keep also in mind is for any of these viruses, there is no cure in infected vineyards, and there is no chemical control of virus vectors that is highly efficient. However, control of vectors can be of use, particularly in the case of Leafroll, to mitigate the impact of Leafroll and reduce its spread. So, keeping in mind that there is no cure in infected vineyards for any of these viruses, it is highly critical to contemplate the idea of being very selective at sourcing the planting material. And the planting material should be tested for viruses and shown to test negative for these viruses. That is the best method to prevent the presence of viruses in vineyards. And this is where the National Clean Plant Network, and through its efforts at producing clean vines and maintaining clean vines in foundations collections, can be is actually of tremendous help. And obviously, the combination of efforts through the National Clean Plant Network with state certification programs is really paramount to limit the presence and dissemination of viruses in propagation material. And with this, I think I'm just going to close and pass the mic back to Chrislyn. Thank you very much for your attention.
All right, Marc, thank you. We do have questions, a pretty good amount. The first one is from Daniel, who asked, do we know yet whether red blotch symptoms are a good indicator of virus titer in the plant, and whether symptomatic vines are more likely to facilitate transmission of the virus than vines with latent infections?
Daniel, thanks for the question, which is really excellent. We do not have information to see whether there is an association between virus titer and efficiency of transmission by a vector, let's say the 3 Cornell alpha-alpha 3 hopper. However, based on our understanding as to how other grape viruses move, I would not personally suspect any association, meaning that a symptomatic or an asymptomatic infection would be equally efficient for the vector to acquire the virus and move it. But that's, a personal intuition more than anything else.
Okay. And then 2 related questions from Tim and Margaret. Tim asked, what is your sense of how widespread red blotch is in the eastern production areas? And then Margaret asked the same question, but about Leafroll.
Well, it is my experience that Leafroll is really widespread and very likely explained by the fact that when the Eastern industry expanded their vineyards dramatically several decades ago, clean material was not available in sufficient amount to supply the demand. And that is very likely how Leafroll became so disseminated. In terms of red blotch, my understanding is red blotch can be widespread in terms of, and the relationship there is the origin of the source of the planting material. My experience indicates that if material is sourced locally, the likelihood that vineyards are established with red blotch is much reduced compared to material sourced from outside of the Northeast.
Okay. Let's see here. Then David asked, are the symptoms of Leafroll and red blotch as pronounced or distinctive in red hybrids as in vinifera?
Very often, that's another excellent question. Very often in hybrids would be, the infection of Leafroll would be latent in hybrids, meaning the virus would infect the hybrids, but very little symptomatology or none actually would be expressed. The vines would not manifest typical symptoms. However, they can be infected and they can serve as a source of the virus for mealybugs and soft scales to move them and start a new infection somewhere else, whether in the same vineyard or in an adjacent or closely distantly related vineyard. In terms of red blotch, I personally only have experience with Chamberson, and Chamberson does actually shows some reddening of the leaf blade.
Okay. And then this question from Suzanne is kind of related to that, so I'll go ahead and ask it now. She said, Leafroll is symptomless in hybrids. Is there anything known about whether or not it still affects bricks? And is there any information on red blotch in hybrids?
Excellent question, Susan. Thanks for asking it. In terms of red blotch, I'm not aware of any study that is underway to evaluate the effect of red blotch on vigor, yield, or fruit quality. In the case of Leafroll, there have been studies in the past that have documented the effect of Leafroll on hybrids, showing a reduced sugar content of diseased fruits, whereas other studies have shown little to no effect. So I would say the jury is still out there, but keep in mind again that the effect of a virus is a result of the interaction of a given rootstock sign combination and the virus strain in a given environment. So there are so many factors that will influence whether a virus is impactful or the degree of it being impactful, and this can vary from location to location and site to site. But otherwise, it doesn't mean that if you don't see symptoms that there will not be any yield or quality depression.
Okay. And then this is also somewhat related. Patrick asked, do any species of vitis have resistance or immunity to these viruses?
Another excellent question, Patrick. To my knowledge, there is no cultivated or wild grape, meaning vitis, regardless if it's a European grape or a hybrid or a native, that is known to be resistant to a virus, unfortunately.
Yes.
Therefore, breeding programs are basically not an option to contemplate the development of resistant cultivars.
Yeah, resistance is different from symptomless.
Yes, tolerance versus resistance. And to my knowledge, there is no resistant material. However, there is, coming back to tomato ringspot and the dagger nematode, the effect of tomato ringspot is much more pronounced on own-rooted, hybrids, as we have shown Chelois and Vidal during this presentation. And just grafting the Chelois or Vidal will enable a grower to get a crop, just because these rootstocks are tolerant to the nematode vector.
Okay, and then one last question from Rick, who asked if a grower plants clean certified vines in or near a leafroll infected vineyard known to harbor mealybug, what, if any, control measures are recommended to control the spread of leafroll to the clean vines?
Excellent, Rick. And yes, it is very key that if you establish a vineyard with clean material, you keep on scouting, because mealybugs, can be dispersed by, strong winds or equipment that move from vineyard to vineyard. So our recommendation is to keep scouting, and if you see any vine that becomes suspiciously infected, to remove it as quickly as possible to contain the spread of the virus. And although Movento has been shown as an excellent chemistry against mealybugs by reducing the population size by close to 90%, if not more, I'm always very reluctant to recommend the use of Movento because we haven't documented, its efficacy in terms of control and protecting clean vineyards. However, this is an option that people have the liberty to select.
Okay, thanks, Marc. I think it's time for us to move on to Mizuho's presentation. Mizuho, I have your slides pulled up and have given you control, so if you want to unmute your microphone and go ahead. All right.
All right, Chrislyn, thank you very much. Can you hear me okay?
Yes, just fine.
Excellent. All right, so well, first of all, thank you, Tim, for inviting me for this webinar. So Mike gave us a really good overview of these viruses. So what I'm going to talk about today is about what's going on in Virginia, especially in the field. So as Marc indicated, the symptom of leafroll particularly is more pronounced on the berries, which you can see on this slide, the one on the center with a nice symptom on the leaves, and then also discoloration on the skins is shown as an example here. So I'm not going to go too much talk about Leafroll virus itself, but I think I have to change these numbers now to 60 to 70. But we have found a good amount of Leafroll in our region. So Marc also talked about transmissions. I'm not going to go into the detail, but one thing I want to mention about here is the size of mealybugs. As Marc mentioned, it's really tiny, and then they tend to hide under the bark. So when you're scouting for mealybugs, you really have to spend the time to peel the bark off and then look for them really actively. So in terms of species we found in our vineyard in Virginia, we have two mealybug species we found. One is grape mealybugs, which Marc also showed the picture of it. The other one is gilt mealybugs, which is shown in the top of the screen here. It's a little bit hairy and a little bit larger than the grape mealybugs, but if you ask me, I cannot really tell the difference between them. It's so hard to distinguish the difference between mealybug species. This gilt mealybugs is known to be a pest for pistachio and almond. And I don't know why they're doing their activity in the vineyard in Virginia, but they are pretty widespread. And then my student Taylor Jones is working on a transmission assay, and it looks like it's just fine to do transmission of Leafroll 3 from infected vine to the healthy vines. And also that the mealybugs not only be able to transmit virus, but they can also cause their issues by producing honeydews, and then these honeydews is basically going to be a food source for the other fungal diseases such as sooty mold, so it can be an issue as well. So when I joined the Virginia Tech back in 2009, we didn't know much what's going on in terms of Leafroll or any other virus diseases, even though Marc helped us in the earlier days to check on the presence of Leafroll 3 in particular vineyard. So what I did was conducting a survey for about 4 years. And then you can see the map of Virginia there, and then you can see that the flags. These are the vineyard I visited to collect samples. So in total, I visited 77 vineyards, and then they have different blocks in the vineyard. So as a total, we had about 415 samples processed, and then which included 39 wine grape varieties. And also, while we are on the road, we collected about 100 wild grape samples, which could be within the field or up in the mountain or just next to the highway. So what we found was about 8% infection with Leafroll 2, 25% infection with the Leafroll 3, and then only 1% infection with the grapevine flake virus. So the reason why we decided to go with the grapevine flake virus was that they have a literature showing that if they have a mixed infection between Leafroll 3 and the grapevine flake virus, the effect of it to the vine is really greater than the individual infection. But looks like, fortunately, we don't have to worry too much about grapevine flake virus in Virginia. So what it means by the 25% of sample being infected by Leafroll 3, if I recalculate based on the vineyard I visited, it will translate into 64% of vineyard contain at least one infected vine sample in their vineyards in their block. So it looks like they have a fairly high infestation rate among our vineyard. That would be our concern. And then luckily, we didn't find any of these 3 viruses in our wild grape samples, which doesn't mean that they cannot go in there though. In California, they found that the Leafroll 2 in their native species, British California. So one of the things we are wondering about it is that there were so much differences in terms of the percentage of the infection among our samples. So what we did was we collected other information such as variety, age, and then location information, and then presence or absence of mealybugs to see that if we can actually tell what's going on from our data. So first thing we observed was a difference among variety. So one thing we can tell was that none of the cultivar was immune to the Leafroll 3 infection, as Marc indicated. So these are the subset of our samples, and then only cultivar we didn't find infection was Chardonnay, which I misspelled in here. But the issue with this data set was that the Chardonnay, we only have 5 vines or 5 samples. So lack of infection here is probably more like an artifact of lack of sample size. The other thing I should mention here is that, as Marc indicated, that American grapes and then also French American hybrids, they tend to not to show any symptoms in terms of Leafroll disease. But from our survey, then they have a very high percentage of infection, which is shown in these 2 arrows in this slide. So they could harbor these Leafroll viruses and not showing any symptoms. Looks like it is. Oops, here you go. All right. The other factors we examined was that the effect of age. So oftentimes we couldn't get good information on the ages of the vine, so we decided to go with very general categories. So what we decided to do was dividing our samples into 3 age groups. One is planted before 1990, second one is planted during the 90s, and then third one is planting during the 2000s. And you can see that the sample size in here in the second column. And then in terms of Leafroll 2, what we found here is that the vines planted before 1990s had a significantly higher percentage of Leafroll 2 found among their samples. In terms of Leafroll 3, there the incremental increase among these groups, and then these are different from each other, significantly different from each other. And then, of course, the vine planted before 1990s had a really high percentage. So one of the reasons we speculate here is that the reason why we're here today, probably because of the effort of the National Clean Plant Network and then Foundation Planting Service, which probably kicked in the mid-90s. The result from of the Leafroll 2 is shown here in our data set that the rate of having infection is much lower after that people start obtaining the certified material. And then, of course, the certification program itself has been improved over the last decades or so. And then in terms of Leafroll 3, since we have a vector in the field, longer that the vines in the field, they probably had a higher probability of being infected by through the mealybugs or scale insect. Right. The other factors we examined were the symptoms. Once again, Marc mentioned about symptoms is really not a reliable measurement. It is shown as the data in here too. So for both Leafroll 2 and 3, symptom basically doesn't tell us much. So they don't have any association of having symptom or not. One of the reasons why is due to the fact that they have other diseases such as red blotch virus could show the same kind of symptoms, or other disorders such as nutrient deficiencies. Also, lack of symptom doesn't guarantee you that they don't have virus in it. And then in terms of mealybugs, we found about 11% of our field being infested with mealybugs. And then when mealybugs was present in the field, they have a higher probability of having Leafroll 3. And then with Leafroll 2, since they are not vectored by mealybugs, the relationship was not really strong. They don't have any the significant relationship between presence of mealybugs and then presence of the Leafroll 2. All right, and then what we also did was that we tried to see that is there any differences among the regions we have in Virginia. When we did that, we didn't find any effect of regional differences. However, what we found was that if we start adding the factors in terms of geographic levels, the more information we found in a smaller spatial scale, it's better for the model to explain the source of variation. What it meant was that probably the transmission of Leafroll 3 by mealybugs is happening in a fairly small spatial scale, not in a regional scale, of course, but we're talking about vine-to-vine movement. And then to indicate the same kind of the movement of the virus or a tendency of having that infection, we also examined the aggregation of the Leafroll 3 infected vines. And then the younger the vineyard, the tendency is that we are finding more aggregation of the vines being infected by Leafroll 3. But as vines getting older, the aggregation disappear and it's become more uniform. As you can imagine that the mealybugs moving virus around among the vines. And this is one of the cases where we monitored one of the vineyards over the course of 3 years. So you can tell that in 2010, they have about 8 vines infested with Leafroll 3. And then by 2011, we have more than 30. So one point to make here is that the mealybugs can spread the virus rather quickly. And then also, once the vine was infected with the virus, they can be infected for the life. So they have a continuity in terms of the time, but also that the mealybugs seems to be spread vine to vine based on this data. And then the other thing I should mention here is that the orientation of the movement. So it was basically following the rows, indicating that it's probably following the movement of the people in this case, because we didn't use any airbrush sprayer in this field. We are using backpack sprayer, but the people are moving along the row to do the maintenance of the vines, such as the canopy management and so on. So probably these tiny insects carried by the human and then just moved around. All right, so based on the information from the field, that it looks like a mealybugs is pretty important in our environment. We conducted 2 types of field trials to examine the efficacy of insecticide options. And then a first type of trial was the prevention trial. I have a few field trials, but I don't have time to go over everything, so I'm going to give you a few summaries of it. But this particular field was infested Cabernet Sauvignon vines more than 20 years old. And what we did was we removed everything but one vine in the panel. And then we replanted with certified Cabernet Franc vine. It was planted at 5 and 10 foot from the old vine. And then, so idea here is that to having the new vines interplanted within the same panel, can we see that the movement of the mealybugs or movement of the virus, the new vine, and if so, how quickly it can be done. And also on top of that, what we did was having the insecticide treatment. So the first treatment we tried was a sale at bud break, and the second treatment is a sale at the bud break, and then followed up by basaloid at bloom, and the third one is a check, which didn't receive any insecticide. We have 3 years of data, but I'm going to just show you the 1 year. So we have 3 panels of graphs in here, and then first one is untreated check. So you can see that the x-axis here is the number of insect count, and you can see that the spike of the mealybugs count in end of the June 30th. When we did an application of insecticide at bud break, then the peak seems to be moved about 2 weeks to the July 15th. And interestingly, when we had an application of the insecticide at the bud break and then up bloom, then we have a spike of mealybugs instead of a decrease in the mealybugs. So our speculation here is since the application of the insecticide at bloom, which is shown on the arrow in terms of time, was the broad spectrum contact insecticide basaloid. So chances are it probably killed off all the beneficial insect we had in that field. And then mealybugs, as I mentioned, they tend to hide under the bark. So they are probably protected by the physical barrier. And then when they came out, they didn't have any natural enemy to worry about. So they end up having a really good time in this particular case. And then the other thing I should mention here is that the spread of the Leafroll 3. So 2009, which was the first year when we planted the certified vine, after 6 months we tested these vines, and then one newly planted vine was already positive with the Leafroll 3. And then in 2010, we start noticing that the mealybugs moved to the younger planting. And then 2011, 31% of the young vines were infected, and then, which was regardless of the treatment, as you can imagine, based on this graph. The other type of trials we are conducting is more like a rescue operation where we know they have an infestation of mealybugs, and then can we do something about it by applying that insecticide. So in this particular case, we tested two different systemic material. One is a Movento, which Marc mentioned, and also Scorpion, which is a neonicotinoid. And then, of course, we have an untreated check to compare. In this particular case, we have two applications at bloom and then 2 weeks after that first application. And then we repeated 2 years to see that the effect of this treatment. So the first year was 2011. As in the last slide, the y-axis here is the count of mealybugs, and then x-axis here is the dates that we measured or counted the mealybugs. So in 2011, there was a general decline of population of mealybugs across all treatment. But when we compare the number of count of mealybugs, both Movento and Scorpion significantly reduced the number of mealybugs compared with the untreated check. In 2012, the population in general was much lower than 2011, which was not only the case with this particular vineyard, but also the same with other trials we conducted in quite distant vineyards. When we compare that mealybugs count, Movento was significantly lower than Scorpion, and then Scorpion was significantly lower than the check. So as Marc mentioned, it does work fairly well if you just monitoring that count of mealybugs. Then other thing I should mention here is that it looks like that they have at least 2 spikes of mealybugs increase, one at around mid-May, and then the other one at around mid-July. And it seems to be fairly consistent across the years and location that to have an increase in mealybugs just around the bloom and then about a month after that bloom. And then sometimes we found that the tiny spike at the end of the season as well. So it looks like at least they have a 2-generation increase within the season. The other measurement we did on this particular treatment was that we count mealybugs before we apply treatment. And then all the vines were treated with the same material over the 2 years. So when we count the mealybugs at the beginning, in 2011, of course, we didn't find any difference among them. Then in 2012, after the first year of treatment, we found that the Movento, which is a spirotetramat, numerically lower than the other two treatment, was not significantly different from other two. But in 2013, we found a significantly lower number of mealybugs at the beginning of the season before any treatment application with Movento than the other two treatments. So it looks like they have a little bit of evidence of carryover effect of Movento, which is understandable because it is one of the material it actually goes up and down in the vine compared with the dinoteflon or scorpion, which only goes upwards in the vine. All right, so the take-home message here is that among three viruses we surveyed is already there in different degree, but we found more of the Leafroll 3. And then, as I mentioned, looks like clean plant material seems to be a key to the management. And then vectors are also common in Virginia. And then there are movement of viruses. And then Leafroll 3 can spread rather rapidly throughout the field. And then I didn't have a time to mention other trials, but we did have other trials to do test systemic materials such as Movento, Scorpion, and Rosebud. However, Rosebud is not labeled for that application, a foliar application, so it's not recommended anyway. But this material can provide a good control if you just count the mealybugs. However, as Marc mentioned, we are not able to completely stop the movement of a virus going in, simply because even though it works it works by killing the mealybugs, but it does take a time from the acquisition of the insecticide to actually kill the insect. In the meantime, they can spread the virus. All right, so I'm going to just really go through a few more slides to give you an update from our recent study. So we decided to go beyond Leafroll 2 and 3, and then I'm not going to go into these slides, but here's a recent update from our survey results. So we actually added about 300 more samples to see that the overview of virus issues in Virginia. So in terms of presence or absence, yes, and we can find all the viruses we tested so far. So Leafroll 1, 2, 3, 4s are there in Virginia, and then Leafroll 3 is still high in the percentage. It's about 23%. We also found that the respiratory stem pitting virus, which is a part of the root growth wood complex, which is pretty high, but which was expected because that it was not on the list of the virus screening for while. And then grapevine A and B is also present in the field, but it's not in a high percentage. Flake virus, we found a little bit more, but not much. All right, this is going to be the last slide. So to our surprise, we didn't find much tomato ring spot much. We found a little bit, but not in a great amount. Grapevine, the Pinot Gris virus, and also vein clearing virus, we didn't find any in our samples. But red blotch, we found quite a bit. We found about 22%. All right, that's it from me. As usual, I'm the one talking, but they have a lot of people who's working behind, and then Taylor Jones, who's actually hiding their face in this picture, is the one who worked lots in this project.
All right. Thank you, Mizuho. We do have a couple of questions. Patrick asked if the overwintering form of mealybug is killed by cold temperatures in northern vineyards. And his thought process was that maybe then removing snow around the trunks would help control it.
Yeah, that's a really good question. Based on what we found in the field, probably it's not, our winter is not cold enough to really kill mealybugs, at least in the Virginia. It's probably depending on the species too. Maybe that's why that we found the Gills mealybugs, but probably not in the New York or Pennsylvania. But
okay.
My gut feeling is they can probably survive fine.
Yeah. And then Suzanne asked, is there a concern of Movento traces being found in the fruit?
That's a really good question. I'm not the expert on it, so I have no clear answer for it. That's a really good question, though. But since they passed the EPA regulation, I'm guessing that they shouldn't find any trace Movento in the fruit.
Okay. And then Tim commented, there's a surprising amount of red blotch. Is there any relation to more recently planted vineyards versus older vineyards?
Yeah, this is a really update that I just got from my student. So I haven't really done the testing like I did for that Leafroll yet, but I have a suspicion that there's going to be a correlation between the age and then a percentage of red blotch we found.
All right. Well, thanks, Mizuho. Let's see here. That was all of your questions, so I'm going to go ahead and switch to Annemiek's slides and let her have control so she can give her presentation. Annemiek?
Yes, thank you very much, and I hope everybody can hear me just fine. First of all, I do want to thank Tim and Chrislyn for putting on this webinar. It's wonderful to be part of it, and I'm learning a lot from both Marc and Mizuho. I just want to talk a little bit about our situation in Michigan. Since we didn't have a grape or virologist working on grapes in the past, we didn't think we had any problems. But of course, at some point, we had to face the symptoms that we couldn't identify. And so we have done some surveys, diagnostic surveys, primarily trying to identify symptoms and understand what viruses we have present in Michigan. And this is obviously collaborative work together with Jerry Gillette, my technician, Naidu, Rayapati, and Basu Bhagawati in Washington State, Solomon Andeshop, Pat Murat, Paolo Sabatini from the viticulture program here, and also Keith Mason and Rufus Isaacs from entomology, and then our extension agents, Diane Brown, Brad Boffman, and Duke Elsner, and the grape growers, and several sources of funding. So the surveys we've done actually since 2010 have been primarily looking at symptomatic grapevines and using ELISA as well as PCR. That was mostly done by Naidu in Washington State. With the PCR, we've done ELISA testing for a number of viruses. And as you can see, out of almost 400 samples that we've had over the years, we found a lot of grapevine Leafroll-associated viruses. The 429 is a combined test. And as you can see, the Leafroll-associated virus 3 was the most common, and a total of 68% of the positive samples had those viruses. We also found quite a bit of tomato ring spot and tobacco ring spot, actually primarily tobacco ring spot, which are the nematode-transmitted viruses. We also have peach rosette mosaic virus, primarily in some of our older Concord vineyards, and then grapevine fleck virus and a suspected sample of grapevine fanleaf virus. So out of the symptomatic samples we had, 42% were actually turned out to be positive. That still means that we had a number of samples, whether they were, whatever symptoms they were expressing, that we didn't get any positives out. So just as an example, the symptoms that we observed for grapevine leaf roll were in the white varieties, the typical rolling of the leaves, and in the red varieties, the reddening, as Marc also showed. The tomato and tobacco ring spots decline is actually the more damaging virus in terms of survival of the vines. I think in combination with our harsh winters, we are actually having vines dying substantially in some of these vineyards. Obviously, the short shoots, mosaic leaves, and also reduced fruit sets, small clusters we observe as well. And then we have a number of symptoms that we weren't able to explain. This is a Syrah vineyard. We suspected that we have Syrah decline, but we weren't able to test for that. Sometimes there's crown gall at the base, so we look for crown gall first when we see reddening of one part of the vine or the whole vine. And there could be other problems as well, mechanical damage and such. So we obviously have some other things going on, and perhaps we have red blotch. We've done some testing, but not widespread for red blotch, so we don't know if we have it. We did hear from a Wilbur Ellis rep that they had a positive sample, but we weren't able to trace that. They wouldn't tell us where it was, and so we couldn't confirm that we had red blotch in Michigan, but we believe that we also have it, so some more testing is necessary. So I just want to share some results where we're looking at the effects of leafroll, since that was so common in our vines, one of the most visible and common problems. And this is a study that was done in collaboration with Solomon Undershaw and Paolo Sabatini, looking at the effect of leafroll virus 3 infection on Cabernet Franc in 2012. And on the left column is healthy grapevines, and on the right, symptomatic grapevines. And the biggest differences really are in Brix level, which I think reflects what was found in New York, about a 2-degree reduction in Brix in this case. The pH wasn't significantly different, nor was the titratable acidity. The sugar per vine was significantly reduced, and no significant differences in anthocyanin and phenolics in this particular year. I should say that 2012 was a very warm year. We had a very warm, hot summer, so even the grapevines that were affected probably had a chance to get them as ripe as they could. One of the other, there were many more measurements done, but one of the things that was interesting is that the net photosynthesis was really reduced in relation to the symptom severity. So the stronger the symptoms, the more the photosynthesis was reduced. We then also wanted to look at a vineyard. This is a Chardonnay vineyard in Southwest Michigan, where there was a lot of leafroll. In fact, about 7 or 8 years ago, they first started to see symptoms, and the vineyard owner mentioned that they had a big and fast increase in symptom expression in the vines. It seemed to really spread. And there's also tobacco ring spot in the same vineyard. So we were trying to look at the effects of these diseases, primarily leafroll, but then in combination with tobacco ring spot on yield and also fruit composition. So these were some typical symptoms. Again, on the left side, you'll see the leafroll symptoms. On the right, the tobacco ring spot type symptoms. Sometimes, obviously, they were combined. And what we found is that actually a high percentage of the vines, especially in the latter stages of the study, almost 95% of the vines were infected with leafroll, where a lower percentage was infected with tobacco ring spot virus. And looking at, this is a map of the vineyard. On the left side, it would be the leafroll by symptom expression, so from no symptoms to very severe leafroll symptoms. And then the middle column shows the rows with tobacco ring spots, no tobacco ring spot, and dead or missing vines. Actually, in this vineyard, about 25% of the vines had died at some point. There were replants in some locations, but those replants also were infected pretty quickly. And then the rightmost map shows the combination, and the purple ones are the vines that were showing both leafroll and tobacco ring spot symptoms. Of course, the symptom expression didn't always indicate the infection. I think with the tobacco ring spot, we were pretty close at being able to recognize the symptoms, but a lot of the vines had the leafroll virus in them, even if they weren't showing strong symptoms or symptoms at all. I just wanted to mention one more thing, is that you can see some clustering of the vines. Actually, we did notice that there's an area sort of a little bit up from the middle where there were a lot of tobacco ring spot infected vines, and obviously a lot of death of vines and very much stunting where we saw fewer symptoms of leafroll. So looking at the effects on yield, we divided vines into categories from apparently healthy to very severe, and then also very severe plus tobacco ring spot symptoms. And what we saw in 2013 was a fairly normal year in terms of production. We didn't see a big effect until we started to see severe symptoms. And then the very severe and the very severe plus tobacco ring spot had upwards of an 80% yield reduction. In 2014, after we had a very harsh winter and a lot of winter kill or winter injury of the vines, you can see the average yield per vine. We didn't reach even 1 kilo, so 2 pounds per vine of fruit. It was actually very difficult to find vines to do the study with, and so you seem to see a spike in the mild vines, but those were vines that actually had a lot of regrowth from the base, and so we might have had differences in yield just because it was difficult to find vines to work with. But again, we see a big reduction in the severe, very severe, and very severe plus tobacco ring spots symptoms. So the combination, we had very low yields in those vines. And then again, 2015, we had again a very harsh winter, but the vines seem to have come through it a little bit better. And here we did see a reduction at the mild, moderate, and severe. But again, similar trends, just showing quite a yield reduction, especially when we have more severe vines, severely symptomatic vines. And the yield loss was a function of the number of clusters per vine, so there was a strong correlation, but also the size of the clusters and the berry size. But really the biggest effect was the number of clusters per vine were reduced. Looking then at effects on bricks, we found something similar as we found before, about a 2 to 3 degree reduction in bricks, especially in the severe category. When we got to very severe vines, symptomatic vines, we actually had so few clusters that perhaps they were still able to ripen them, so we had a little bit less of a reduction in the bricks level, but pretty similar to what was found before, so maybe maximum amount of 3 degrees bricks. And looking at the pH, a slight reduction, and the titratable acids, but this was not as strong. And we did also have mealybugs in this vineyard, and working with the Rufus Isaacs, we have seen that there were quite a few mealybugs in the vineyard, and that actually, unbeknownst to us, there were a lot of vineyards in the southwest Michigan that have mealybugs infestation. So when we started looking, and especially looking under the bark, we found quite a bit of evidence of mealybugs. You can see the white fuzzy symptoms or signs of mealybugs, and we also found some live mealybugs. And up to now, we didn't believe that we had any mealybugs up north. I mean, obviously, southwest Michigan is a lot warmer than the north, but we have also found mealybugs up north. And so this is a big concern, of course, to the growers. They are reaching for Movento in this case, and they believe that they will solve the problems with Movento. But as Mizuho has shown, that we need more recommendations to deal with, especially when we replant vineyards or plant new vineyards next to these old vineyards where mealybugs are very common. So I just want to conclude then that viruses are a threat to Michigan wine grape production. We also have viruses in juice grapes, especially Leafroll. We have found them in juice grapes, but as far as symptoms and actually effects on yields, the wine grapes are the ones that we have most problems with. We do need a comprehensive statewide survey to accurately assess virus prevalence and risk in Michigan, especially now we should also look at red blotch as well as some other viruses that have been found in the Midwest to see if we actually have those in Michigan and what the actual impact is. We do need management recommendations for grapevine leafroll and ringspot decline. Both of them are challenging. I would say the ringspot decline is even more difficult because we're dealing with a soil-borne issue. Those nematodes can live a long time, and so we have difficulty other than fumigation. If people don't want to fumigate, then we need to come up with other means of managing those diseases. But through the surveys that we've done, we have increased awareness of the risks of virus infection and the need for clean planting material. And I think one of our biggest questions from the growers is where do we get that material? Obviously, there's many nurseries out there, and they may or may not have virus-tested material available of specific clones that growers want. But I think altogether we have a positive trend in awareness in Michigan. So I want to stop right here, and thank you very much for your attention.
All right, thanks, Annemiek. We do have a few questions. The first one is Tim, and he asked, regarding tobacco ring spot, what about rootstocks, and would it prevent the tobacco ring spot virus?
Well, I believe the nematode-resistant rootstocks would hopefully be efficient as well in really preventing the nematodes from feeding on the roots or transmitting the virus to the roots. So that is one thing that we need to look at as well in practice in the field. Some preliminary trials that were done by Tom Zabadol with table grapes and grafted table grapes seem to work. So we're hopeful that that would also be effective with tobacco ring spot virus.
Okay, and then Margaret asked if you surveyed the nematode level in the tobacco ring spot virus vineyards.
Okay, good question. Thanks for that. Well, we have looked, anytime we find tobacco or tomato ring spot virus, we do usually test a soil sample also for nematodes, and I think in all cases we have found dagger nematodes. The numbers, I guess, you don't need very many dagger nematodes to get transmission. So even if we find one, I think we see that there's a risk. So, but they're pretty common in our Michigan soils, and we have a lot of sandy soils which are conducive to nematodes.
Okay, well that just answered David's question, who asked, is soil type important in avoiding nematodes, sandy versus clay? So
yes.
All right. And then Tim also asked, do you have any grafted hybrid grapevines? Grafting works here for Vidal Blanc, for example.
That's a good question. I haven't heard of it, but that is definitely something to find out. Yes, yeah, thanks for that question.
That would work for the tomato ring spot virus, or we found it to work. Yep.
Great, thanks.
Okay, and then Marc asked if the Concord vines infected with peach rosette mosaic virus are showing symptoms.
Yes, they are. We have this sort of weepy posture of the vines. Again, you have shortened inner nodes, zigzag type inner nodes, small clusters, uneven ripening. Yes, they do show symptoms. Okay.
Marc, if you are still on, we have a question that's left over from your webinar. Are you still there? Oh, wait. Here's a question from Suzanne real quick. What, if anything, is the difference between tomato and tobacco ring spot virus?
Well, other than sort of virus-specific virus characteristics, not that much. I think they do have a little bit different host range in terms of weeds, and maybe Marc would be able to answer that better. But for practical purposes, we consider them to be very similar.
Marc, do you want to comment on that difference between tomato and tobacco ring spot virus?
Annemiek summed it very well. Basically, they are little cousins of each other, but they are very identical in the type of impact they have on grapes, the type of approaches that need to be used to manage them.
Okay. All right. And then the question that was left over from your presentation was from Deb, and she asked, does weed management help to reduce red blotch movement within the vineyard by suppressing hoppers?
I'm sorry, could you just repeat?
Yeah, does weed management help to reduce red blotch movement within the vineyard by suppressing leaf hoppers?
Leaf hoppers, first of all, if I mention leaf hoppers, I have to apologize because that was not my intent. Okay. Leaf hoppers are not known to be involved in the spread of red blotch whatsoever. As we speak today, there is no evidence that any leaf hoppers can move red blotch in a vineyard. However, there is evidence that the tree hopper, the alfalfa tree hopper, is capable of transmitting red blotch. The question of the weeds and their involvement in the epidemiology of red blotch is a very, very important one. And as we speak, we do not know the answer. However, the potential for particularly cover crops to facilitate the movement of the hopper and therefore the transmission of the virus is a important issue that needs to be addressed very urgently. And I wouldn't be surprised that some of the cover crops might facilitate the transmission of red blotch.
All right. Well, that's all the questions. So thank you to everybody. Thank you very much, especially to Marc, Mizuho, and Annemiek for your presentations. These are great, very informative. Also, thanks to the audience for, number one, joining on a Tuesday after our technical problems on Thursday, and for your great questions. It's always great to have questions from the audience, especially when it's slightly more difficult to ask them in a situation like this versus a live presentation. So thanks, everyone. Tim, would you like to cover this acknowledgement slide, or would you like me to?
Sure. I would like to reiterate my thanks to Marc, Annemiek, and Mizuho for joining us, and for joining us twice since we couldn't connect, and Mizuho in particular because he's still on a visit to Japan, so he is joining us from across the planet. And I also want to acknowledge the other support in our other speakers that are going to be in the series. And we have 2 more coming up, one next Thursday, and then the final one will be a week from Thursday. But I also want to acknowledge the funding support from the USDA APHIS National Clean Plant Network and from the New York State Department of Agriculture Markets, the Specialty Crop Block Grant that helped support this webinar series. And thanks also to the audience, those of you for joining us for an hour and 15 minutes, and we hope you'll be back for the next one. So I'm going to pass it over to Chrislyn.
All right, I think I have a couple more slides here. Come on, let me see. Oh yeah, just basically what's coming up next. This coming Thursday, we're going to have 2 speakers, Tom Burr from Cornell University and Shadi Atala from the University of New Hampshire. Tom's going to be talking about crown gall, and Shadi is going to be looking at more of the economic side of things and the value of virus-resistant plant material. Then Thursday next week, we're going to be talking about New York's revitalized grapevine certification program. Marc will be back. We'll also have Margaret Kelly from the New York Department of Agri Markets, and then we're going to have 3 vineyard or nursery owners from New York State. We'll have Dennis Rack, Eric Amberg, and Fred Murwath. So with that, I think we are done, and thanks again, everyone. We really appreciate all of you.